All right, hi everybody. Good morning again. Thanks for waking up and making it here. As Bertrand mentioned, Joshua can't make it this morning, so I'm filling in for him. I did my master's with Josh at Johns Hopkins and worked on some of this stuff, but it's been a little while, so please forgive me if I'm a little rusty. So what I'm going to be talking about to you today is Connectome coding. And this sounds like a very buzzwordy phrase, so I'm going to start by explaining a little bit about what it is, then talk kind of how we do it and why do we care. And again, many of these examples have been lifted from one of Joshua's talks, which you can find online. The URL might be a little small, but I'll post these slides publicly later. So, as I said, we're going to start with kind of a bit of background, how do we do it, and then what are some applications and how we can use this. So, before we talk about Connectome coding, I just quickly want to talk about a simpler case, which can be neural coding. So neural coding is kind of the act of trying to characterize the relationship between some neural activity and the environment. So in other words, you apply some stimulus and you perceive a response. And trying to understand the relationship between these is kind of this action. So then in the context of connectomes, rather than necessarily the ongoing environment, what we're trying to characterize our connectome in the context of is the history of the individual. So this can be history such as disease, it can be genetics, age, all sorts of things like that. So imagine we have, in the case, again, of neural coding, this spike train. So each of these vertical lines just represents a neural spike. And you can see there, and on the, kind of as they go across the slide, it's moving in time. So as you can see, they're kind of maybe random-ish. There's a lot more at the beginning, but there's also a couple bursts at the end. So these are following some form of distribution. So the goal that we're trying to do is understand, again, the relationship between some stimulus that's applied and the reaction that we get. So essentially, the first step of this could be perceived as the encoding. So again, the probability of reaction given your stimulus. Now the decoding step is trying to go the other direction. We have the probability of some stimulus given our reaction. So the neural code can kind of be summarized as the joint probability of this stimulus and reaction environment. So the simplest way we can, so let's just take the probability of reaction independently. The simplest way we can do this is just by assuming our spikes are independent. So then our spikes, again, don't care about which spikes came before them, and we can say that they'll spike at some arbitrary rate lambda, which means that our probability of reaction is just following a Poisson distribution with parameter lambda, and so on. So this is the type of approach we're going to be taking in the context of connectomes, and we're going to, again, of course, get a little more complicated. So first up, I just want to make sure that everybody's on the same page about what I mean when I say connectome. So by connectome, I mean a graph. And where we have a graph is defined by a set of vertices or nodes and edges or connections. So we have the blue dots on the screen labeled with letters that are vertices, and they're in, the set of big V vertices, and edges are simply relationships between them. So this can be represented as a matrix, as so, where essentially the intersection of a row and a column is an edge between those two nodes. So if we can look at, say, node A, we'd go to the first row, we notice that there's a connection between D and F. We have a red box in columns D and F. So this is kind of more what a connectome actually is. It's not necessarily just a graph that is some funny-looking shape, but it actually has some structure to it. So here we have, again, nodes that are laid out in the brain. We have connections that can be within hemisphere, across hemisphere, potentially within, again, lobes or other subgroups. Some connections that could be homotopic, so reaching to the same regions across hemispheres and so on. So these are the types of networks that we're actually trying to characterize and understand. And again, when you convert this to an adjacency matrix, it looks something like this, where in this representation, I believe, white means edge and black means not edge. So again, to mimic what we had earlier with neural coding, connectome encoding is simply the probability of observing some graph given some history. I left the history variable to be s just for the sake of simplicity. And then decoding is the, again, probability of identifying some history given the graph you observe. And this is kind of going back to So what Janina was talking about with like patient classification, trying to identify disease, what's the probability of some diagnosis given the brain network we're looking at? So then again, the Connectome code, as it were, is the joint distribution of the two. So again, we need all of these 4 pieces to compute these things. We need probability of observing a graph, the probability of some stimulus or history or disease, again, and then the conditional probabilities. So again, the simplest model we can do is assume that every edge is independent, and the probability of some edge between nodes u and v is just p. So again, this is pretty much every edge, again, as I said, is Bernoulli with probability p. So this gives us a connectome that looks like that, which is very realistic, I think. That's probably what my brain looks like, and we can go home. Except that, not really. So as we saw before, there's actually a lot more structure to human connectomes than that, and in part because of how we generate them. So vertices, rather than just arbitrary locations, they can be brain regions, they can be voxels. In the case of EEG, they could be, say, electrodes. In the case of electron microscopy data, they could be segmented neurons, and so on. And then edges could then be physical connections in these modalities, or they could be functional correlations across them. So again, the edges may not actually be independent of one another, and there may be some structure that kind of links them. So, a slightly more complex way that we can represent this is through something called a stochastic block model. And what this allows us to do is define a dependence between edges based on group membership. So we have some probability of connection of edges, again, within a group, some probability across groups. And there's no reason that these probabilities, say, within group A and within group B, need to necessarily be the same. So the PWs on the diagonal could be, so for instance, PW1 and PW2. They don't have to be the same. So you essentially say, okay, so if I'm in group A, I will talk to another group A neuron with probability, again, PW1, and if I'm talking to one in the other group, I'll talk to it with PB, which is, again, in the case of perhaps hemisphere structures we saw below, a lower probability. And then all we need now is some tau vector, which tells us where the regions live. So this, again, can be represented as now a stochastic block model with this block matrix, as we call it, and tau. So these blocks don't necessarily need to just be hemispheres, as I showed in the kind of previous slide. So this is just doing an eigendecomposition of some connectomes and displaying the eigenvectors corresponding to descending eigenvalues. And we can see that their values are often kind of clustered by lobes that just so happen. So we have the second dimension here which is our top figure. Well, it doesn't come up very well in the middle slide, or on the middle screen, sorry about that. Is hemisphere, then we kind of have the frontal lobe in the next, and then kind of frontal and parietal in the last. So then you can impose these, or you can impose or learn these types of this type of block structure, and again, estimate probability of connection within and across these groups, and then use these to identify outliers from populations. So again, the purpose of this symposium is to generate insights from rich, complex, and messy data. So obviously, these graphs, I think I've hopefully convinced you, are rather complex and rich, and they're certainly messy. But how can we actually generate insights from them? So what we can do is try and learn these so-called latent structure models, which means essentially that we don't have to impose, again, this block membership. What instead we can do is say the probability of an edge between two groups, between two nodes, is conditional on on the distance between two feature vectors in some arbitrary feature space. Again, whether it's behavioral information that we've collected from our patients or something else entirely. What we're trying to do is just, again, come up with a feature vector that's informative for representing the connectivity between nodes. So what this allows us to do is to define some subset of nodes, edges, and structure, and then learn others. So again, this can be very, very, very useful if we, again, say estimate connectomes for a bunch of participants. We've imposed the node structure so that they're all aligned and we can compare them. We've, again, estimated edges for each of them, and then we're trying to learn structural parameters of these networks. Okay. So now for some applications. So One perhaps obvious application is simply trying to understand the difference in structures between functional and structural connectomes in humans. So this is, on the top we see structural connectomes from diffusion MRI, on the bottom we see functional connectomes from fMRI. And so again, the left column is simply just an example connectome from each of these types. And on the right column, what you see is are essentially probability distributions or histograms of on the top you see the ipsilateral connections as compared to the contralateral connections. So to summarize that figure, you see that diffusion connectomes consistently have higher ipsilateral connectivity from contralateral. And again, this may seem obvious. Things that are closer will have higher probability of connection. So then, in the fMRI case, for example, you can see that what we have actually is much more significant homotopic connectivity. So in other words, communication or essentially syncing between the same regions across hemispheres. So these are some simple types of analyses we can do to explore. So another application that I'm currently working on with a colleague of mine at McGill, and there's a poster on the poster floor that you can check it out, is trying to use connectomes to validate a parcellation that was generated from PET data in trying to in Alzheimer's disease. So what Jake has done is trying to parcellate the hippocampus based on early and late tau progression regions. So then what we can do is we can impose this structure on our graph and again see if there's differences in terms of edge probabilities and distributions between them. Another application that I'm working on with Stephanie Suarez and Bratislav Misic at McGill is trying to essentially form a link between different types of connectomes. So what you see on the top is an illustration that shows that we have some structural connectivity matrices. We feed them into a deep net, and we're trying to predict functional connectivity matrices on the other side. So the performance is the graph on the bottom. You notice we have a few different groups. The average across all of them is 0.63 performance. And so one of the first questions that kind of jumps out from this figure, I think, is, okay, so why is there this separation from, let's say, the BNU1, 3, HNU1, and SW4 data sets and HCP and Lausanne? So one thing that we did here was we actually used different edge weighting between these different groups and wanted to see if that had an impact. And also, we didn't process them all with the identical pipeline. So those on the left are actually processed with a pipeline that I'll talk about in a little bit called NDMG. And the others were processed with other pipelines. So we're actually in the process of kind of extending this work and going the other direction as well, where we're predicting structural connectomes from functional connectomes so that we can learn the structure that, again, can link these networks. And we expect that we'll be able to get similar or increased performance simply because, as you may recall from an earlier slide, functional connectomes are much more dense. So as I mentioned, I would quickly talk about NDMG. So there's a paper on bioRxiv and is currently in submission that kind of talks about some of these analyses and this pipeline that we use to generate all the connectomes in the paper. Please check it out and give feedback on hypothesis or something like that so we can make it better. And I'd just like to thank everyone who has helped, whether with figures, science, insights, or what have you. And that's it.